in these data sets. So this is really what we think is the QLattice is so good at. It's this really strong feature selection part. It's the finding interactions between features and then building these simple models that generalize well on new data sets. So it's not only the predictive power on unseen data, but it's also understanding why the prediction is happening. That's so strong in this case. If I just take this a bit further here, let's lay out the landscape in machine learning. It might look something like this, where you plot prediction accuracy of a certain method versus interpretability of that method, where you have these 4 quadrants. So the top left, you might have the high performing ensemble type of methods like random forests. And on the bottom right, where interpretability is high, but prediction accuracy is not the best, you might have your linear models and decision trees. Just to show what's being done currently, at least from my view, this is a paper from very recently, where a group has found a biomarker that is very strongly predictive for Alzheimer's disease. Not only that, they also combine it with other clinical metadata. And they do a very good job at this, but it's a sort of a laborious task by manually fitting linear models and step by step building the features you need to make your prediction. It's very interpretable, but you could argue you lose a bit of performance. So when you want to hunt for more performance, then of course you would try something more like a random forest. And this is another recent publication on some skin indication, skin disease dermatitis here, where they applied a random forest to some sort of omics data set, and they found a set of features that's predictive. But here the struggle is to understand why the prediction is happening. And okay, you have your set of features that seem to be contributing to the signal, but you have no idea how they're interacting. What is a possible mechanism here, right? So it's really this relationship between proteins, between genes, or any other entity that you have in your data set, where the QLattice can add a lot. So we would like to think of ourselves in this top right part, where we have high prediction accuracy while maintaining interpretability. That's a very bold claim. And certainly we can talk in detail about what this actually means. But with the next 2 use cases, we will show you that this is actually this holds true in at least in our hands. What Lüge and Valdemar will show you now is sort of a typical workflow with the QLattice and the data scientist type of person. We have a training data set that we feed into the QLattice. We generate a set of graph-based models that can also be expressed as mathematical expressions. And then Lüge and Valdemar will talk you through sort of a mock workflow. How do you explore these models? What do you get out of them? Right? And just to show you what we are currently working in together with big pharma and smaller companies, as well as universities. So we are very big success in various RNA medicine applications, predicting toxicity, stability, things like efficacy. Again, because we not only predict very accurately why a certain RNA drug works or is not toxic, but we can also tell the scientists why, which then helps them to design better drugs. Secondly, chemoinformatics seems to be a suitable field, especially if you allow the QLattice to use features that you can explain. So, of course, it would be more challenging to do this on bit encoded features, even though that also works. But where it really shines is when you have features that you can actually explain, something like the log P, lipophilicity, the whatever. Is it acidic? Is it basic compound? What does that do to my binding activity? Whether it can cross the blood-brain barrier, or whether it's lipophilic or not. So pharmacophores are, of course, a very interesting one here. And then clinical research, we also have good use cases with biomarkers, real-world evidence, and multi-omics. We'll actually show you multi-omics in a second as well. And I think at this point, I'll hand it over to Libby.
just go one slide ahead if you can.
Oh, yeah, a second.
Yeah, thank you, Sam. So I will give you an example from drug discovery. And I'm Lykke, and I also, as Sam said, started at Abzu just recently. And I come from a background in biophysics and bioinformatics, and have been part of the drug discovery in Roche for several years, mostly in the field of antisense oligonucleotides. And that's also what I will be exemplifying here for you. And I've taken these antisense oligos all the way from early discovery until the preclinical stage of the first entry into human. And what I will showcase to you today is the therapeutic potential of oligonucleotides that does splice modulations. So what is splicing? To give a chance for everyone to maybe understand the concept. So you have the DNA in every single cell that is then being transcribed into RNA. And RNA consists of exons. These are the colored boxes here. And the gray ones, that's the introns. And doing splicing, what is happening is that each exon is spliced together. You could say they're glued together. But in the cell, it can happen that some exons are not glued together, but others are. And that's what we call alternative splicing. And from that, you actually get different proteins, which is depicted down here. So for example, protein A contains all the exons. Protein B, you can see that exon 3 is left out. And protein C, their exon 4 is left out. So it's different proteins. They are different kind of proteins. Then there's different kind of diseases where we know that there are different mutations in different exons, and that causes the disease. For example, in Duchenne muscular dystrophy, it is known that there are mutations in certain exons that, that turns the dystrophin gene into a non-functional protein. And then there's another disease called spinal muscular atrophy, where you actually want to include an exon because it will substitute one protein. And actually for the spinal muscular atrophy, there is actually a drug on the market called Spinraza. You may have heard about it, where you actually go in and modulate the splicing in these patients. And before treatment, in the most severe cases, these patients would die at an age of 2, and now they will survive longer. So I will now share my screen. Can you share? Because what I wanted to show you now is how we actually work with these kind of data in a Jupyter notebook. So you should see. So again, what we will be looking at here are skipping of exons in the dystrophin gene to treat Duchenne muscular dystrophy. And in Duchenne muscular dystrophy, you have mutations in the gene DMD that codes for the dystrophin protein. And that's important for proper muscle function. And you can have mutations all across the gene, and the gene is actually very long. It contains 79 exons. And what we'll be looking at today is mostly where you have mutations, where you have a deletion of exon 55, or 50, not 55, 50. And when you do not have exon 50, you will have an out-of-frame mRNA, which means that you will have no protein. You will have no dystrophin protein. But what you can do, if you could skip this exon 51, you would actually have an in-frame. You would have shorter dystrophin protein, but you would have an in-frame mRNA and a functional dystrophin protein. And that's what is depicted down here. So if you could skip it, you will have an in-frame mRNA and you will have a truncated form of the dystrophin protein. So down here, what is depicted is we have these ESE. That's exon splicing enhances. And if you can block these, you will probably skip the exon 51. But I say probably because it's not just straight away that you put an antisense oligonucleotide, which does just simple base pair. So ACTG base pairing with the mRNA. That's just not how it works, because there are many different factors that actually turns these antisense oligonucleotides into a proper drug. So they have drug properties. First of all, they need to be active. So what we have looked at is a data set that contains of 155 splice switching oligonucleotide SSOs. You can also call them AONs, ASOs. They have many different kind of names. And they are being classified as effective if you have exon skipping of at least 5%. In the paper, there's several different features by which they annotate the SSOs. And they have actually up to 50 features. Some of them could, for example, be what are the number of the nucleotides A, C, U, G? What percentages of these are there? What are the predicted exonic splicing enhancers? So these were these parts of the exon where you see that the splicing factors can bind and induce exon inclusion. And if you block them, then the exon will be skipped. You could also look at the secondary structure of the SSO. That's what I've depicted down here. So it can actually fold upon itself. It can also form two identical SSO, can also form duplicates. That's what's depicted here. And for those structures, you can calculate the minimum free energy. In the paper, what they do a Wilcoxon rank-sum test applied on all these kind of features, and then they see if there's a significant difference between effective and ineffective SSOs based on these features. They also do a PCA to try to look at what features could potentially discriminate effective and ineffective SSOs. They pin down 4 different features, mostly around the ESE predictions, about a splicing factor, TRA2 beta, something about the binding energy between the SSO and the target, and then again, another splicing factor, SC35. So they classify the SSOs effective and ineffective based on these 4 features, and actually obtained quite a well, you could say they have pretty good recall, as we would call it. That's the true positive rate. But as you can see, when they try to predict the ineffective, it's almost a coin flip whether they actually make it or not. So could we maybe use the QLattice to see if we have this set of up to 50 features, could we pin down some of the same features as they have found in the paper? But instead of doing the very laborious work of going through all the Wilcoxon rank-sum test and the PCA and the manual looking at all the data, can we use the QLattice? So as Kevin said, the feyn library, that's how we interact with QLattice. So we start by importing that and other Python packages that I will be using. We load in the data. The data is here. So you have 156 compounds and almost 50 features. So we can scroll around here and see all of them. And then out, that's whether it's skipping or not. A 1 means that it's an effective compound and 0 ineffective. We split it up in a train and test where we leave 1/3 for the test and 2/3 for the training. And we have just around 70 effective compounds and 35 ineffective compounds in the training set. So what I'm doing now is that I'm connecting to the QLattice and I'm clearing all the learnings of the QLattice. Next, I'm setting up, I'm getting a QGraph from the QLattice. And the QGraph is just a container of all the graphs where you have the input register, so the features, and the output, the thing that you want to predict. In this case, it's a classifier. You could also make a regressor in the same way. And there are actually 2 categorical columns in the data set. And I need to say that these 2, they are categoricals. So I'll put that in. So what I now have is a list of graphs. You can see here that the loss is not a number, so it doesn't exist because I haven't fitted the data yet to all these. So what I will now do is that I fit the data to the graph, and then all these graphs up here are sorting according to the laws. And what I'm doing is I'm using the binary cross entropy because it's a classification. And as you can see here, what comes out is that it pins down the rescue ESE, the same as in the paper, and the TRA2 values, the same as in the paper. And it goes into an ad and makes the classification of whether it's an effective or ineffective. We can then actually inspect this graph a bit more. We can look at it. So the numbers above each of these nodes, that's the Pearson correlation to the output. So we can see here that the hexameres from the rescue, that's hexameres that are present in these ESE sites. And the TRA2, that's this SR protein, that's a splicing factor, you could call an SR protein. And the metric for the train set is over here, and for the test set, now it's cut off. It became a bit smaller now. I hope you can still see it. But you can see that we actually generalized pretty well from the train to the test. We have a pretty high recall as in the paper. The precision is less. That's the same as in the paper. So now we have a simple model. It has actually pinned down, the QLattice have pinned down on the two factors that also in the paper were shown to be important. And the neat thing is here that we have two features. We can write it up as a simple mathematical equation. Because it's a classification, we have this log-rec function before. We can plot the ROC curves between the train set and the test set, and then lie quite tightly, which means that we generalize pretty well. We can also plot the confusion matrix. Now I've done it for the whole complete data set. And from that, we could calculate the metric for the full data set and compare it to what they have found in the paper. So they have an accuracy of almost 80. For the complete, we have 72. In the paper, it should be said that they don't split between train and test. They do the whole classification on the whole data set. So let's try and interpret this graph. We can do that very simple by simply plotting, do a partial plot, as we call it. So what we will have here is that we have the number of these rescue ESE hexameres, and then the predicted output. So if it's a 0, it's an ineffective compound. If it's 1, then it's an effective compound. The colored line here are increasing value of the TRA2 beta. And we can see for very low values, we actually seem to have almost all the compounds are predicted as effective. But as that number goes down, and as the hexameres go down, we actually predict the compound to be ineffective. But we can, if we have compounds with a low TRA2 value, we can try and see if we can increase the hexamer count. Put it simply at a different position on the exon, and then it could become more effective. But we can see in general here, the most promising one is actually if you just find spots where you have these rescue ESE hexameres, then you will have an effective compound. And that's exactly what they also write in the paper, that the hexameres is important, and they are actually surprised about the inverse correlation with the TRA2 value with the effective. So the higher the TRA2 value, the less effective compounds. So this was a simple graph. Then let's try and see if we could go even higher. So what I'm doing now is that I'm allowing the QGraph to search for even deeper graph. So now you can see that it now allows to have a depth of 2. And we see that it still pins down on the TRA2 value and the rescue ESE as before. But now it also has the binding energy between the splice switching oligonucleotides. And now it also included the total number of A nucleotides. And as it goes through these loops, fitting the data to the long list of graphs, sorting them by loss, and then going back and updating the Q graphs with the best performing ones, we see that the loss goes down, and essentially, and in the end, it actually latches on to this model. Now it doesn't change as much. What is changing now is mostly the loss. So the fit becomes better and better. Then what now happens when we look at the metrics for this? So we can see, if we compare it to the previous one, which we have here, that the accuracy has gone up for the train test. But for the test set, it hasn't gone up. It's actually decreased. So it doesn't generalize as well, but it performs very well on the train. So do we still see the same interaction between, we had this, that if we had a lot of hexamer count, the SSOs would be predicted to be effective. And if we had high TRA2 values, they would be ineffective. We should still be able to see that. And luckily enough, we do. So high numbers of hexamers give you an effective compound. And if you look at the TRA2 values, so the low numbers, that's the blue and purple, purple, pink, magenta, these two lines. And if you have higher values, which is this 20, it's this line here. So we still see the fact that we have, for higher values of TRA2, we have less effective oligos. But still, there's also a dependency on the binding energy between the SSOs and also the total number of A's. We could have actually inspect this even more. We could do a partial plot in 2D where we plot the binding energies and the total number of A's against each other. And the colors are now that the more the pink color, then it's ineffective, and the green, then it's predicted to be effective. And if we can see here that if we actually put the TRA value too high and the hexamers too low, that was where it could be difficult to actually design effective compounds. Then if we actually have a high number of A nucleotides and also a high binding energy, then we can actually design effective splice switching oligos, even though we have low TRA2 values, high TRA2 values and low hexamer counts. So this is a way to actually guide. If I were to design the splice switching oligos, I would now know that I would need to go for target sites with a high hexamer count and with a low TRA2 value. But if that's not possible, sometimes the RNA simply have a composition where it's not possible, then I would know, well, then at least I need to go for places with high number of A's. And I also need to increase the binding energy between the compounds. And the binding energy you can actually also increase just by doing more on the chemistry side to actually modify the nucleotides. So that was it from the example with the splice switching oligos. So any questions?
Not many hands. They're saving all the good questions for later.
Yeah.
So I'll continue from here. And we're going to switch gears. Once I have my PowerPoint back in control, somehow it's not doing what I wanted to do. There we go. The other use case we wanted to show you is a multi-omics case. So we've been exploring various data sets, including a couple of omics data sets, and we have a few on our webpage where you can go through a tutorial and understand what the QLattice is doing with very wide data sets with many features and few samples. We have a cancer diagnosis data set that does very well with 2 features, and we have an Alzheimer's data set that we can also do very well on. But then what I wanted to explore further is how does the QLattice explore multi-omics cases where you have more than one data type that you put together into your data to make a prediction about your target variable. And there are not that many multi-omics datasets out there currently. So the most interesting, or let's say comprehensive I found, was a TCGA breast cancer dataset that had a couple of data types that we used. So just to briefly introduce the use case. It is a data set of 705 breast tumor samples. And these samples were grouped into invasive lobular carcinoma and invasive ductal carcinoma. On the right-hand side, you can see the difference in various biomarkers and also the location of the tumors. And there were over 600 patients here who survived, and 95 patients who died eventually from this cancers. And we wanted to train a classification model on whether we can reveal some of the mechanisms underlying breast cancer severity. So it's a very, let's say, still a shallow research project, but it shows great potential, and it might inspire some of you to try this on your omics data. More detail on the actual data. So we have copy number variations here. We have somatic mutations, we have gene expression, and we have protein expression. And what does it all mean for the people who don't work with this data? So most of you will know this central dogma of, we start from a genome, that's DNA, that is transcribed to mRNA, messenger RNA, that then gets translated into proteins, which are the machines in the body that do all the hard work. And it's on these various levels where things can go wrong, and where there's information to be gathered and integrated. So the first two I mentioned here, copy number variations and somatic mutations, they lie on the DNA level. So copy number variations are essentially just varying numbers of gene copies in different people's genomes. If you have more gene copies, it's often the case that you have higher expression of that gene, leading to higher protein levels often. And if you have less, it's the opposite. So it's a proxy for gene expression in a sense. Then you have somatic mutations that are sort of usually more sort of local changes in the bases in your genome. It could be a single nucleotide polymorphism, like a SNP thing. It could be a deletion. It could be a frame shift where suddenly you're missing something that messes up your translation and transcription. Just to give you a quick introduction to that. And in this case, we actually only labeled whether a gene had a mutation or not. We didn't include the precise information on the mutation. So essentially, this is a binary variable. Various copy number variations goes from -2 to +2, meaning I think it's something like in the average population, people have 0, and then if you have more than usual, you have positive values, and if you have less than usual, you have negative values of that copy number in that gene. Then we have gene expression. So this is on the mRNA level here. It's essentially each mRNA is a subset of your gene, right? So it's copied from DNA into RNA, and it holds information for the protein later on. And gene expression is a proxy for protein expression in a sense. So, and then we have protein expression measurements as well. And since this is quite an old case, we are quite limited in the measurements here. So this is reverse phase protein assay. So not a high throughput mass spec method or Olink or anything like that, but it adds some sort of information. And we included that in this multi-omics prediction as well. So in total, we'll end up with something like 1900 features that we will feed into the QLattice and ask the QLattice to build us simple models that are also highly predictive. So at this point, I'll hand over to Valdemar. Do you want to share your screen?
Okay, am I on now? Great. Okay, you can see me. I'm Valdemar. I'm a data scientist in Abzu, doing hands-on projects. So I'm looking at a lot of different data and have been working on this Multi-Omics case together with Sam as well. Before jumping into this case, I have these 3 takeaways that if you're going to bring anything home today from this session, I hope these 3 will be it. So what we bring to the table here is interpretability, and that's the interpretability of the graph, but also of the mathematical formula that you're served. That's like the raw model that you can actually go and inspect, and that's kind of leads to the simplicity of what we're doing. We are really boiling down stuff to the raw core of the signal in your data, and we do that by doing heavy feature selection here and very intelligent feature selection. Hopefully, as I will show you. But with that said, interpretability, simple models, and feature selection. Oh, let's jump into the breast cancer case. What I've done here is that I have poured training data into the QLattice. I have had it running for 1 or 2 minutes, and I've come up with a list of all the best graphs that we've come up with at that time. What I've also done is that I've constrained the QLattice to only provide me graphs with 2 features. And this is the way I typically work with the QLattice to try to set like a lower bar of simplicity. So how much can I actually do? How much signal can I extract with only 2 features? That's the question that I first put forward, and that is what you're seeing here as well. So scanning through these 2,000 features, the QLattice gives me these 2 features: 2 gene expressions, ApoB and MYOC, together in a Gaussian relationship can actually go ahead and predict the severity of these breast cancer cases. That's what the graph here tells me. Translated to the mathematical formula down here, what you see is the logistic function on top here that is converting what you could call the model into a predicted probability. That is the actual output. What you also see is the Gaussian, 2-legged Gaussian in action here, where you recognize the gene expression of ApoB together with its parameters. That is what we've, through stochastic gradient descent, have found to be the right parameters. And the same setup over here with the RS-MYOC gene expression. So this is actually not a multi-omics output, you could say, since we're only looking at gene expressions. But the QLattice doesn't really care. You could say it just picks up the features that tends to have the highest, the best prediction power with only 2 features. So from here, let's go and see how we perform. But first things first, what I like to do is to try to understand, okay, so what happened here? What did the QLattice do? And one simple way to do that with a 2-feature model is to just do the scatter plot, really. So what you're seeing here is the RS-MYOC, the gene expression. It goes from 0 to 16. And similarly, the RS-ApoB. And we have all our samples here. And then we have the vital status up here. So 0 means that survival, and 1 means that you died. We see an obvious cluster here. So my guess would be that the QLattice has found the hotspot, the Gaussian up here, and that will point to this area being of high importance when it comes to predicting severity in these breast cancer cases. But let's see. First, I'd just like to have a look at the performances here. On the train, we're seeing an AUC of 62. That's kind of on the lower end of the scale, so there's not that much signal in this particular mechanism that we've pointed to. And then similarly, an AUC of 65 on the test set. And also, as Lykke noted, this is like an interesting part about working with the QLattice, that you very often experience that you go ahead and actually like generalize perfectly, you can say, with these simple models. So it's kind of a pleasure to work with in that sense. One thing to perhaps note here, and to capture some questions perhaps, is what would a standard ensemble model do? So I've just ran this on a random forest as well, and I've received similar AUCs as what you're having here. So a random forest model with 2,000 features, how would that perform? How would that generalize? It ends out actually generalizing as well as a 2-feature model, easily inspectable, right? So that's like our value add. Okay, so but actually kind of lifting the curtain here a little bit for what the QLattice actually did. Here we are looking at this partial plot that you came to know from Lykke's presentation as well. Again, you're seeing the RSM-RC on the x-axis and the ApoB on the y-axis. So in a way, it's the same plot as before. You have the dots being the different individuals, and again, you see the green ones being the people that died. And then you have kind of the background here actually explaining the model dynamic here. So this is the Gaussian hotspot, as you see, that performs this decision boundary and leaves that as the model that we're using. So that is how easy you can go from 2,000 features, 2-minute run on the QLattice, having a model, 2 features that brings you something that competes with like the machine learning field in general. But this was starting out very simple. As Lykke did, I also went ahead and allowed for more complexity, because what you really want to do is to try to find this complexity frontier, you can say, to figure out, well, this data that I have right now, is that of very complex signal, or is it fairly easy signal, so to say? So that's why it's a good idea to start simple and then move up from there. See, does my AUCs follow with my complexity, or am I just as well off with a simple model? That is what I often find, that I can come a long way with a simple model. Here we're picking up other types of omics. So we're picking up a copy number, and we're picking up a mutation. So TNXB is thrown in the mix as well. And then we don't have the MYOC. We found another gene expression that actually fits better into this feature cocktail here. And again, you're served with a mathematical formula. It's directly translatable from the graph, of course. So what you see is 2 additions and then the multiply. Again, you have the logistic function, and then up here you have your model up there. So you can recognize the 2 additions in the parentheses and then the multiplication across the 2. Okay, moving on here. Again, trying to digest. Okay, so what just happened there? I'm doing one scatter plot here with the gene expressions. So the gene of VWDE against the ApoB, which, as you might have noticed, was the same one as before. So that was not out of luck, you could say. ApoB definitely has a say in which cancer patients is more likely to survive. And again, you see some sort of cluster up here. So my guess is that the QLattice has picked up some decision boundary up here. Although this is not a 2-feature model, so we are actually dealing with we're dealing with 4-dimensional model, right? So it's not enough to just be looking at one scatter plot here, as I will show with partial plots afterwards. But just to actually also go ahead and look at the performance metrics here, we've reached an AUC of 0.7 here, and a test result of 0.74. So with the more complex model, we are generalizing even better. So it seems as if that the rising complexity really added some signal in this model here. Let's go ahead and try to dig in and get a feel of what's going on here. And we do that with what is now partial plotting, since we are looking at specific, you you can say, settings in the model. So you have to go in and put in fixed values of some of the variables to get a feel of what the model is doing. But that is what we're looking at here. Again, we are keeping the RS VWDE in the bottom and the ApoB on the y-axis. But the mix here is that we have to go ahead and put in the fixed values. So over here we have mutation carriers of the TNXB gene, set to 1, so these are bits. And then over here we have the non-carriers of this gene, of this mutation, sorry. So we're kind of carving in the layers of this scatter plot and lifting some specific observations that we all of a sudden can start designating like that. And that is what you see here. So some of these dots are bigger. Some of the samples are represented by these values. And we go ahead and have a decision boundary that is lower on the RS ApoB. So with having a mutation carrier of TNXB, we say, well, the need you only need a low RS ApoB for us to say you have a high likelihood of not surviving cancer. Being a non-carrier, it's a different picture. So in that case, you would want we would see need to see high values of VWDE together with ApoB for us to go ahead and do a prediction of high severity. So this is how we go ahead and really dig into our mathematical formulas and try to get a feel of what our model is doing. But it really comes down to what is taking place here. So you can really read it all from this, so to say. But we have just made these plots that's available in the feyn package to help you dissect and figure out what does this formula really tell me. Yeah. So I think I will leave it there. I guess you could say for the researcher, this is like a fine first step, right? So you've actually come up with these models or hypotheses, if you will, for you to look into. So what I would recommend the researcher digging in the multi-omics field of breast cancer that would go ahead and try to understand, well, what are these specific features actually doing? Do I understand why they would have a say in this case? And in that way, try to do some kind of contextual validation of what the model is telling me as well. And then this will be the first iteration of that research avenue in this data. And we will leave it there at the first iteration on this case. But hopefully you've been inspired to how such an iteration flow could look like. Yes, that is it from me. Any questions?
We had a couple in the chat.
There is